Good morning. It's my great pleasure to introduce our speaker of today, Dr. Uwe Oelmueller, who is at QIAGEN at the Molecular Diagnostics, leading the sample technology developments, and he is also the coordinator of the Horizon 2020 project SPIDIA4P and convener at CEN and ISO, and he will talk today on standardization of pre-analytical workflows. So please, we're looking forward.
Yeah, thank you very much, Kurt, for the nice introduction. I'm giving this presentation with in my role as the SPIDIA4P coordinator, and you will learn more about SPIDIA4P during the presentation. The title is on new CEN and ISO standards for pre-analytical workflows to improve diagnostics, biobanking, and research. So, why are pre-analytical workflows so important for diagnostics, for research, biobanking? Now the answer lies in some numbers that are just presented here. If we look into deaths of patients and adverse events of patients, we recognize that the diagnostic errors cause about 10% of this patient death and 70% of the adverse events. When we look deeper into this, where do these errors come from? Then the pre-analytical phase is the biggest contributor. So depending on the study we look into, it's between 50 and 70% is the contribution of the pre-analytical phase. If we turn this around, we can also say if we improve the pre-analytical workflows, then this is the biggest lever to improve also the research results and the diagnostics results. This is also associated with costs. So here's one study presented from the US. So a typical US hospital has 650 beds. There's a damage of about $1.2 million per year due to pre-analytical errors. There are studies for Europe as well, for example by Frost and Sullivan, and they come to very similar numbers for Europe as well. But the negative impacts or errors that occur in the pre-analytical phase do not only impact diagnostics, but also research. And one study that is often cited was presented in +1 in 2015. And this paper presents numbers that 50% of all preclinical research is not reproducible in the US pharma industry, causing $28 billion of damage. This is not only caused by pre-analytical impacts, it's about 1/3 that is caused by reference material, lack of standardization, and pre-analytical impacts. But it's high numbers, high damage to patient, but also to healthcare and to research. So why is that? That is meanwhile quite well understood where these errors come from. And therefore I would like to walk you through the whole diagnostic process. And that always starts with the patient. The patient is the first variable, so to say. So the patient can be healthy, the patient can be sick, the patient can have exercised the day before the sample is taken, diagnosis is initiated. So these are first variables. But then there's one important step, and that is a sample collection. And there we have to be aware that we take living cells out of the body of a patient or donor. And these living cells come from a very well-defined conditions in our body, and they are transferred into a collection device, which has a total artificial environment. So the cells come in contact with plastics, they come in contact with rubber, the oxygen supply is different, nutrition supply is different. So for them it's a crisis. And the cells react to this crisis quite fast. For example, by changes in the gene expression, gene upregulations, downregulations, changes of protein profiles, modification of proteins, and changes in metabolite profiles. So easy to explain. Yeah, so they also do this in the body when we get infected or we are injured. Also, their cells react quite fast to this. But the tricky thing is that these living cells continue to change the sample that is taken out of the body as long as they live. So that means during the transportation, during storage, until they arrive in the laboratory. And in the laboratory, then usually the sample is treated somehow, it is lysed, it is then killed. And then these biological changes are stopped. And then usually the analyte that is intended to be examined during the analytical test is isolated and introduced into the laboratory test or analytical test. And then the diagnostic result is generated, which is reported back to the patient. But one main source of errors is that the changes that occur here are changes in the cells, and the assay cannot discriminate anymore whether it is really measured what was in the patient or what was artificially generated during the pre-analytical workflow. So it can lead to a wrong diagnostic result because an artificial pattern or analyte measurement is done which was generated during the pre-analytical workflow. That is one of the main reasons for the wrong results we see. In the laboratory itself, the preparation of the sample, which belongs also to the pre-analytical workflow, there we see more physical, biochemical, chemical impacts, for example, cross-linking if we fix the sample, degradation if we do not manage to get intact nucleic acids, or also loss of parts of the sample during the sample preparation. But these are not biological changes as in the first part. So as a consequence out of these observations that are very clear is that we have to change our view what an analytical diagnostic test is. An analytical diagnostic test is the result of an entire workflow. That is a very important change of view that we all should do, and I will come back to this during my presentation. I only have one data slide to back up these observations, and this is a study, a pan-European ring trial that was run during SPIDIA. And there was one sample taken from one donor in Florence, a central laboratory, and then portions of this sample were sent to almost 100 laboratories in the European countries. And the laboratories had the task not to store the sample longer than 48 hours, either at room temperature or at 2°C Celsius in ADTA tube or stabilized tubes. And those who stored the sample in ADTA tube as room temperature, which is very widespread, after 48 hours, they determined a more than 100-fold higher level of this mRNA than originally was in the donor's body. So it's a more than 100-fold wrong level, which the assay could not mark as being wrong. Even at 2 to 8 degrees, there were big variations compared to the time 0. Only in a stabilized sample, so the profile was maintained. On the right-hand side, you see another transcript that decreases over time. So this is an increase, here's a decrease. But again, after 48 hours, this was not the level as it was in the body, but that's a level generated during the pre-analytical workflow. So it clearly shows if we don't take care for that, we can get wrong diagnostic results and research results. So what do we need to prevent this? Technologies which prevent these changes. That's the easiest way, so to say. But we also need standards as educational tool, harmonizing tools to prevent this error in the healthcare systems or also in research programs. Now, developing these technologies and developing standards, that cannot be done alone by a small group. That's a big effort. So the first initiative we had in Europe was the SPIDIA consortium that was funded under the Framework 7 of the European Union from 2008 to 2013. So 16 partners from academia and industry worked here together with the European Committee for Standardization on 2 topics: to develop new technologies that preserve the samples and prevent the changes for blood and tissues, and first time ever develop new standards for pre-analytical workflows for implementation in the European countries. And I will come back to this with more details during my presentation. Now we have a successor program that is called SPIDIA4P, SPIDIA for Personalized Medicine. And SPIDIA stands for Standardization of Pre-analytical Workflows for in vitro diagnostics. And SPIDIA4P is even bigger. This is 19 partners and 14 other consortia and stakeholder organizations now working together as a big consensus initiative to work on standards. The goal is to develop additional 30 new CEN and ISO standards in addition to the 9 ones we have already, but also to develop the accompanying external quality assurance schemes to control the laboratories if they want that they are compliant and generate good quality samples. And another goal is also to implement this all in the European countries. So there's a website, www.spidia.eu, where more details are presented, and also a newsletter can be subscribed. So everybody of you who would like to have more information than presented in this talk, you can go to this website or also subscribe to the newsletter. So how does standardization work? Now, if we have a European-wide standardization initiative, the basis of everything are data, evidence. And that was generated in SPIDIA and is now generated in SPIDIA4P's partner consortia. In SPIDIA, so we did run 6 pan-European ring trials in all European countries. We did run consortium internal trials with other partners, with US partners also, like the US National Cancer Institute and so on. A big data pool that demonstrated the impact of the pre-analytical workflows on the final result, identified the specific variables and presented also means how to standardize. And with these data, so we went to CEN. CEN is the European Committee for Standardization, and presented these data. And specifically, so within CEN, there's one technical committee, 140, for in vitro diagnostic medical devices. In this committee, all the European countries are represented by the national standard institutes, and the decision was made to take up this work and develop 9 standards for the European Union, technical specifications, which were published and introduced in Europe in 2015. Now, the next step is to go international, outside Europe. And there is an agreement between CEN and ISO that is called the Vienna Agreement. The Vienna Agreement is put in place to avoid that CEN and ISO duplicate work. So in this case, so Europe took the initiative, and the results were presented to the ISO sister committee, which is the ISO Technical Committee 212, Clinical Laboratory Testing in Vitro Diagnostic Test Systems. And this committee said, yes, we want to take up the work that was done in Europe and develop these standards further to international standards. So that's the highest level of standards we can have. That decision was made in 2014. And at the moment, all eight are progressing to have this as a final draft international standard. This is only editorial changes allowed, and then there will be introduced in the member states of this technical committee, and also will be introduced in Europe and will substitute the technical specifications. So that's how the whole standardization community works and how the standardization workflow is up to an international global level. Now, very often the question is why CEN? Yeah, we have professional societies who could also do it. And that's for sure, they have capabilities, but CEN, the European Committee for Standardization, is the only institution in Europe that is recognized by the European Union, which means by the member states, plus associated countries, and the European Free Trade Association as being responsible to develop standards for whole Europe. So it is the right way if we want to have a broad standardization initiative for Europe. And another thing is very important. So because at CEN and ISO, we have many countries, the national standardization committees are there, plus other stakeholder organizations, we can reach a very broad consensus during this process. And that's why openness, transparency, national commitment are important elements in this process for achieving a big impact of these developed standards. And so the 34 European countries are working together there. That means one European standard, if it gets introduced, replaces the 34 individual national standards. So we achieve a harmonization over all Europe. And here it says again, so the technical committee in which SPIDIA representatives are working together with others. So there we have the 34 European countries, but also almost all the major other stakeholder organizations, like the European Commission, European Society for Pathology, European Federation for Laboratory Medicine, etc., etc. And they also take part in this standardization process that they can agree on the final document. So very broad consensus that can only be achieved if we go the way via CEN. Also, the duration is long. It is not an exercise that is done in a few days or weeks. That's why I present here the duration for developing an ISO standard. It's usually between 3 and 4 years until it is ready. It has various rounds of commenting and fine-tuning, again, to achieve a very high level, but also to get consensus by all the stakeholders. So very intensive work until such an ISO document is finished. Now, the role of standards. Yeah, so we all come from a life science area. We are not so much used to it. It's a difference to other areas like engineering, car industry, so on and so on. More used to it. There are two roles. One is the traditional role. So we write a standard when a field is more mature, and then the standard is a source for technical know-how, but it's also a source for legislation, broadly applicable. But there is another role that was demonstrated by studies and is now a core element in the strategy of the European Union in research programs, and the second role is to support innovation. So if we have a new technology until it takes up in the market, let's take a biomarker, new technology to detect the biomarker, validating the biomarker, bringing it into healthcare system can take up to 15, 17 years. Shortening this period means benefits for the patients, but means also economical success for the companies or institutions. Why? Because if we write an early standard, the standard contributes to avoid mistakes during the take-up of the new technology. That's why this is another important role. And startup companies who early on use standards have been proven to be more successful. So another role also for younger fields to be faster successful and support success. Now, I've mentioned that out of SPIDIA we developed 15 technical specifications. These are presented here. So they all address the pre-analytical workflow, which starts with the patient, with the sample collection from the patient, and ends with the isolated analyte. These are the 15 ones for blood. So there's one pre-analytical workflow from human whole blood for isolated cellular RNA, genomic DNA, circulating cell-free DNA, from FFPE tissue, isolated DNA, RNA proteins, frozen tissue for RNA proteins, DNA is in the making, and for metabolomics in urine, serum, and plasma. 8 out of these, with the exception of metabolomics, which is a little bit behind, I mentioned they're all in late stage to progress to full international standards. Now, a brief glimpse what these standards are. And I have here an example. So that is the technical specification for the pre-analytical workflow from whole blood for cell-free circulating DNA. So the whole document goes through all the steps of the diagnostic workflow until the analyte is isolated, so the pre-analytical workflow. So it starts outside the laboratory. So that means when we go to physician, it's outside the laboratory. What is important at this moment? So what do we need to know about the patient, how to collect the sample, how to store it, transport it, where are risks, where are recommendations or requirements. This is all presented here as a frame. It's not an SOP, it is broader, but it sets a frame to improve results and avoid mistakes. And then it continues until the sample reaches the laboratory and then goes through the steps within the laboratory until the analyte is isolated. And again, it gives a frame about the steps, where are risks and where are recommendations and requirements. So it helps the user to establish these workflows. Now, SPIDIA4P, I said this is a successor program. SPIDIA4P intends to broaden this portfolio of standards. And the standards we intend to develop within SPIDIA4P do address the prominently upcoming field of personalized medicine. So they focus on circulating tumor cells, other extracellular components like exosomes in whole blood. It addresses other body fluids like urine, saliva, stool, final aspirates. Metabolomics will progress to international standard. It's now a technical specification. And for FFP tissue, for in situ stainings, the pre-analytical steps for the in situ stainings. And I mentioned that already, SPIDIA4P will also work on implementation of these standards, mainly in Europe, but also internationally. It's another important goal. And to prove once more that small companies who early on use the standards are more successful. So Inivata is one of the startup companies we have in the consortium. They're growing very fast and have very early on decided to let themselves accredit and use standards. Now, when we developed these standards, originally the target audience was diagnostics. But within CEN, I mentioned that, we have a broad stakeholder community. And there was an immediate discussion whether we should only restrict the standards to the diagnostic field. And very early on there was an agreement, no, this would be too short if we only limit the standards to diagnostics. We should also address biobanks, biomedical translational research with these standards. Why is that? Now, if a new diagnostic test is developed, that means there is a target, an analyte target, a biomarker, for example, and this biomarker needs to be developed. It needs to, scientific validity has to be shown, the assay has to be developed, and for all of these steps, we need samples. Where do these samples come from? From the biobanks. And then these samples are used in research to develop a diagnostic test, which then is introduced into a healthcare system. That means it makes a lot of sense that the standards we develop are used in all of these steps. Because otherwise, if the biobanks would have different standards, we would have a breakage from the biobanks to research. If we use other standards in research, we have a breakage to diagnostics. It does not make much sense if you want to standardize. So there was an agreement with the stakeholders on ISO level, on CEN level, with the professional societies and infrastructures. Now we write these standards also for biobanks research and diagnostics in order to have the same standardization in this workflow of discovering biomarkers and developing the diagnostics for it. Now we have another element which we did not know when we started SPIDIA. There is a new law, a new regulation in Europe since 2017. It is called the European In Vitro Diagnostic Regulation in this case. There's also a new one for medical devices, but this one for this area I'm talking about, it's the In Vitro Diagnostic Regulation. And this regulation, which has the same wording in all European countries, has prominently in there the workflow thoughts, which I have presented when we develop an in vitro diagnostic medical device. And has prominently in the sample quality and the pre-analytical workflows. And I've just copied here one paragraph. There are other parts in this law that also address these pre-analytical workflows, but this is just one. So from the Annex 2, and just you see here, this section shall describe the different specimen types that can be analyzed, including their stability such as storage, transport conditions, time frame between taking the sample, its analysis, against storage conditions, duration, temperature limits, even up to 3 thaw cycles. So these are exactly the pre-analytical variables that we also have discovered in SPIDIA. So we find now a law that takes this up. Very important support also for what we are doing, because it now is mandatory. And now it generates a very broad picture where we are in. So we have a law, European In Vitro Diagnostic Regulation, which has pre-analytical workflow parameters in the law. Now we need translators, documents that help the users to translate the law into their laboratories. The law itself has so-called harmonized standards, which are usually horizontal, broad standards, but also ours that we have developed, which are specific workflow standards, so-called vertical standards. They can help if they are used by the laboratories to follow the requirements and recommendations in this law. So they have now much broader meaning than we initially thought when we started this. You remember, so we started with a huge problem because the pre-analytical workflows were not standardized at all. And now we have this very nice framework, law, standards. And now below the standards, the laboratory within this frame, because I mentioned these documents are a frame. And within this frame, then the laboratory can develop specific SOPs for their very specific application, which then needs to be supported by technology and products. And you remember initially what I said. So in order to solve the whole problem, we need technologies, at the very bottom, and we need standards, which are the upper levels. So very nice picture for us, and underlying again how important it is what SPIDIA4P and all the consortium partners are doing here. Yeah, and with this, so I would like to thank you all for your attention, but I also would like to thank the SPIDIA4P consortium members, but also all the other partners. Yeah, so they're not on this final slide. So on ISO and CEN and all the professional societies and other stakeholders who are supporting us on this journey, because without a broad supporting community, we would not be able to achieve this all. Thank you.